Hello, and thank you so much to Cambridge Zero for inviting me to talk. I'm really sorry to miss the opportunity to present live, but I'm very glad that I can talk to you about underground networks through this recording. I'm Bethan, and I'm the project manager for global data science at SPUN, which stands for the Society for the Protection of Underground Networks. Before I go into what we do at SPUN and how we are focusing on Cambridge Zero's theme of climate accessibility, I'll briefly explain what we mean by underground networks. So the underground networks that we are interested in at SPUN are, of course, underground fungal networks. Sadly, fungi have received really a fraction of the attention that plants and animals have. And they're an enormous group of organisms. So they're a whole kingdom. For example, you have the animal kingdom with all of the diversity that entails, from worms to elephants. And just like this, the fungal kingdom, it's an entire kingdom of diverse organisms. So you have so many different types of fungi. And just like you have scientists and researchers that specialize in particular groups of animals, like ornithologists who study birds, at SPUN, we have our own group of fungi that we're particularly interested in. And these are the mycorrhizal fungi that form networks in the soil. These mycorrhizal networks really underpin terrestrial life on Earth. And this maybe sounds like a big claim, but really the organisms that you see in a natural environment, so the trees, the invertebrates, the charismatic animals, they're all supported by the presence of an underground fungal network. These mycorrhizal fungi face quite a thankless task because despite their importance in ecosystems and despite the fact that they've been playing this critical role for over 1 billion years, they are hidden from our sight in the soil. So it's quite difficult for us to see and to understand their contributions. Mycorrhizal fungi form symbiotic associations with plants. This means that really they physically live inside the root systems of their plant partners, and they provide them with nutrients from the soil in exchange for carbon that plants produce during photosynthesis. Fungi are like us because they need to eat the products of photosynthesis, essentially, that plants make. But these mycorrhizal fungi don't do it by farming plants like we do. They do it by living in very close contact with plants and trading with them. So this is an image of different plants that are interconnected by hyphal networks. These are mycorrhizal networks. I took this image from a recent New York Times article that was produced about SPUN, and I really encourage everybody to look into this because it was a really great article. It's a very nice demonstration of the mycorrhizal symbiosis because it shows how these networks of really fine fungal threads, which are shown in purple here, they link together different plant species in the soil. These mycorrhizal fungi are excellent foragers. They're actually better than the plant root systems really are. They help plants to explore the soil for water and nutrients, sometimes even to a point where the plants actually get a majority of their nutrients and water through their fungal partners and not through their own root foraging capacity. Unfortunately, as is the case with so many organisms right now on Earth, the areas where these essential networks exist are being severely degraded. We're losing so many biomes where these fungi operate and exist. And with their loss, we're really losing crucial components of natural ecosystems. They make up around 50% of the living biomass of the soils where they're present. And it's not just the contribution of these networks to natural systems that we should be considering, as these fungi are also critical to soil health. What's especially concerning are the predictions that the world's topsoil could become unproductive within 60 years if we continue along the rates of degradation that we're currently seeing. A component really of this productivity loss then is the loss of these mycorrhizal networks. And also in industrial agriculture, there's this hugely damaging force to fungal networks and mycorrhizal networks in particular, because it hasn't been designed with these fungi in mind. We're dumping huge amounts of fertilizer on crops in these industrial systems, despite the fact that fungi have this huge impact on the phosphorus uptake capacities of plants. Not only would the loss of these mycorrhizal systems destabilize entire ecosystems, these fungi are also a major carbon sink. So when their plant partners pass them carbon that they have fixed from the atmosphere during photosynthesis, these fungi store it in the soil. When we measure things like deforestation and the impact that it has on climate change by adding carbon back to the atmosphere, we also need to know how much CO2 is being released by the destruction of soil fungal networks, particularly mycorrhizal networks, because they store huge amounts of carbon. This really means that these networks are very, very important factors that should be considered in international climate agendas and reports. Really, another reason we want to understand these networks is just simply the thought that we could lose them before we even really understand their full extent of their contributions to life on Earth. This is where SPUN comes in. SPUN, as I mentioned, stands for the Society for the Protection of Underground Networks, and it's a non-profit science initiative that was founded to map mycorrhizal fungal communities and to really advocate for their protection and conservation. The idea for SPUN originated with Toby Kiers here, who's seen sampling some mycorrhizal networks in Chile. Toby's a professor of evolutionary ecology at the Free University of Amsterdam and has really been working tirelessly to understand how underground mycorrhizal networks operate. And Toby founded SPUN due to this need to conserve these networks as well as to understand them. So we now have a marvelous team of people working towards this aim. We actually have been growing recently, and we have even more amazing team members than are present here in this picture. Luckily for SPUN, there is currently a momentum and interest in fungi and mycorrhiza that may give us the opportunity to overturn what is really an above ground bias that we tend to have when considering the conservation of biodiversity. Conservation initiatives have a tendency really to overlook fungi, and fewer than 600 species of fungi have actually had their conservation status assessed. We know that there are these above ground biodiversity hotspots where we have incredible biological forms and species and interactions, for example, in the Amazon. But where are our examples of underground biodiversity hotspots? So what ecosystems, what regions, what soils contain incredible biodiversity of fungi? Where is the Amazon of the underground? Is it in the Amazon? We don't know. So when we can measure and predict where different fungal networks are concentrated and which mycorrhizal fungi are present, we can then maybe start to predict the way this biodiversity will change under climate change. We would be able to understand what the presence of different fungal species and groups tells us in terms of the functionality of the ecosystem. For example, different mycorrhizal fungi have different traits. So some may be very good at helping their plant partners to survive in drought conditions. Others are particularly good at carbon sequestration. And really knowing which fungi are where will give us a better idea of priorities. So we really want to understand these networks, we want to protect them, but we need to know which networks are in trouble and which ones we need to focus on right now. And so how do we do this? There is plenty of data out there that has been collected by all of the amazing researchers worldwide who have gone out sampling mycorrhizal fungi. Usually this involves taking soil samples from diverse ecosystems and biomes, and they've sequenced the DNA and found in this, the DNA that they found in this soil, and they've used this to identify which fungal species and groups are present. These data are currently available in a database called Global Fungi, and SPUN and our associated scientists have used Global Fungi's data as inputs to machine learning algorithms. These algorithms use not only the data on the fungi that are present across the globe that they've obtained from DNA sequencing, but they also use additional information such as climate variables and the types of vegetation present. And so using all of this, our scientists are able to create maps of predicted below ground mycorrhizal biodiversity. And they've done this for the two main groups of mycorrhizal fungi. This is a prediction map for one of those two main types, which is the ectomycorrhizal fungi. These tend to be located in forests because their preferred hosts are often trees. And you can see in this map here that the predictions of high fungal biodiversity, well, high mycorrhizal biodiversity are indicated by these lighter colors. These maps are really helping us to understand where the hotspots of fungal biodiversity are, but in order to improve them, we need to collect more data. So we can map the underground by taking soil samples and sequencing the DNA there to understand which mycorrhizal networks are present. But the team at SPUN are really not the ones who will be doing most of this. And this is where I'll really address the theme of climate accessibility. As you can see on this map of available data here, this is the available data on underground networks that we have. There's really a lack of data points that have been collected in many regions of the world due to power and funding imbalances that exist in science today. We want to address this problem in a manner that is fair and democratic. So there's been a precedent for this type of global scale sampling project to be run by well-funded researchers, often from Western nations, who will go to countries in the global south and they'll sample the biodiversity there, while providing really little in the way of collaboration with local researchers and local capacity building. At SPUN, we really don't want to operate this way. We are forming a network of researchers across the globe in an imitation of the fungal networks that we study. We want to support mycorrhizal researchers to carry out research in their own countries and biomes of interest, since there are so many amazing scientists out there who are already studying mycorrhizal networks in areas where we have a lack of sequencing data. We want to provide really whatever is necessary for this work to be done. So we are working on public protocols for sampling. This will include protocols for things like DNA extraction from soil and the analysis of this data as well. We really want to be a support network for mycorrhizal researchers. We're also planning to directly fund the sampling of fungal networks. So right now we have a grant program and it has an open call, and we have an emphasis on undersampled regions of the world. We require grant recipients to work with local collaborators in the regions that they're sampling in, and we prioritize local applicants or researchers with sustained collaboration in the study area of their interest. We will also be prioritizing researchers from the global south, as well as women, Black and minority ethnic candidates and other groups that are underrepresented in science. This grant call is also really just a part of our Underground Explorers program, which is aiming to help fund and facilitate research to explore global patterns of mycorrhizal fungi in a fair and democratic manner. So we're also developing a program for citizen scientists. We really hope that the way that science is carried out can be shifted somewhat, particularly the science of biodiversity monitoring. So citizen science programs are amazing and they produce really fantastic results in cases where people are helping to catalogue very visible organisms like birds or mammals. But for fungal networks, so for these mycorrhizal networks, collecting soil samples and sequencing DNA is a little bit more challenging to carry out with a citizen science approach. But these networks are important to all of us, and we want everybody to care and to be involved. And there are exciting developments actually in the ways that people are doing citizen science now. There are developments even in technology like DNA sequencing that will maybe soon make it possible for everybody to be much more involved in these projects. So we want people to form a relationship really with these underground fungi, even though we can't always see them. There will be updates on the Citizen Scientist program in future that SPUN has, but in the meantime, we have a newsletter for everybody to stay updated on what SPUN's doing, and we welcome people to join this through our website. So this brings me on to the final slide. And just this is a huge thanks to the whole SPUN team, not all of which are actually in this image, as well as to our many partners, again, not all of which are displayed here. Our partners and the team have been instrumental in sharing data, producing the prediction maps of mycorrhizal biodiversity, and in working with us on sampling trips and much, much more. And lastly, I'd really encourage everybody to check out our website for how to be more involved with SPUN and to keep up to date with our work. And do have a look at some of the media that has been released recently that focuses on SPUN. And then thank you again to Cambridge Zero for the opportunity to talk. And I will leave my contact details with the organisers just in case anyone has any particular questions or wants to get in contact directly.